Good morning, everyone, and I'd like to thank the organizer for giving me a chance to participate in this very all-inclusive conference. And I'm learning a lot already, and I hope I have something to share with you. And I came from National Institute of Health in the U.S., and very few people from the U.S. are present here, not an EU member, but science has no boundary. And working at NIH for over 20 years, what I found is our workforce is made of 80% or more people not born in the US. So I hope people here will be interested in joining NIH at some point. It's our research group mostly made of postdoc fellows. So whoever is in the White House doesn't affect how we work. So today I'm going to talk to you about one of the specific biological problems and using both crystallography, cryo-EM, and with dynamic modeling as well. So during this pandemic, everyone knows that we get vaccine shot to induce antibody that can recognize as protein of the coronavirus, so we are protected at least from the internal organ overdriving reaction to the virus. So the question really is how our body is so capable of making antibody, any kind of antibody, as long as we expose to antigen. Basically, our genome is limited in terms of size, but our ability to make antibodies unlimited, right? That's really a very interesting biological problem, which is the problem solved in evolution. Our body's antibody is made up, instead of each antibody is individually coded, each antibody is made of heavy chain light chain shown here at the bottom. And it's made of different gene segments, green, red, and blue. And each segment has multiple copies, slightly sequenced different. By randomly selecting one green, one blue, one red, we can make a heavy chain. V and D and J segment. And this random recombination and imprecise joining when the antibody pool is unlimited. And our body just keeps generating new antibodies when it encounters antigens, then they become amplified and become more and more specific through the evolution process. So for people who don't have the ability to make this antibody pool, everything developmental is normal, but we need to be living in an isolated sterile box, like the state. So it's all well. And how does this recombination work? It's carried out by specific recombination enzyme called the recombination, and it recognizes specific DNA sequence called 12 and 23 ISS. And those 23 and 12 ISS protein up at the border line of the V, D, and the J segment, and each of those, each segment is bordered by one type of this recombination signal, either 12 or 23, and the recombination can only occur between 12 and 23. So V will not be joining with V, but V will be joining with D, and D will be joining with J only. And so the two signal are paired by RAG1/RAG2 proteins, so it's very interesting how one protein can recognize a pair of different sequences of DNA. And then the most exciting to me as an enzymologist, structural biologist, is how the enzyme in one active site can carry out two types of reaction. So' strand cleavage is by hydrolysis, and then the second strand cleavage is using the product of the hydrolysis, which is 3' OH, as a nucleophile. Then cleave the second strand. And the end product is the recognition sequence is perfect blend end DNA, and the coding sequence is a hairpin. And the hairpin is wonderful because you can open the hairpin asymmetrically, or symmetrically. That gives you more repertoire for making different antibodies. So that was the talk, that will be topic. First part of the talk is RAG1, RAG2 recognize different DNA, and making cleavage, two different types of cleavage in one active center. So we interested in this project for many years since I was a graduate student, first exposed to BDJ recombination biology in the 1980s. But we didn't have any real approach except cutting protein to pieces, so-called divide and conquer, for 10, 15 years. But in 2000, middle of 2000, we were able to express the protein not in insect cell. Insect cell would produce not good protein because BDJ recombination only occurs in high vertebrates. Insect cell does not do BDJ recombination. The protein expressed in insect cell aggregates, soluble aggregates, low activity. But once we move the expression to Hector-9 cell, which is mammalian cell, then the protein expressed well, and we saw the initial protein structure by selenomat. So selenomat was introduced in Hector-9 cell. Labeling was incomplete, and the protein regular to heterodimer, dimer is over 2,000 residues, so it's fairly large. And the diffracting crystal was like that with selenomat, it was 4.5 Armstrong. So it was using data redundancy, just collect many set data. We were able to generate this really nice map. And the methionine sites are highlighted in pink or red. So model was built and it contained two red one, which was showing blue and the green, and the red two on top of the Y-shaped red one are showing magenta. So it's dimer. And initially we grew crystal with DNA, but in crystal lattice formation, DNA was excreted, was not visible in the crystal. So we have to go through again and screen many DNAs, eventually get the DNA protein complex, which is the step forming hairpin. We provide the linked DNA, so you can see the tilting of the end. Well, I think it's okay. Basically, DNA bound to the protein, and the 12 RSS is shown in light yellow, and the 23 RSS is shown in brown color. And what you see is that the end, which we call the non-member binding protein domain, it's this portion makes this yellow DNA bend very sharply by 150 degrees. But the yellow one doesn't bend very much. And the cleavage center is located in the middle of the Y. So below that is a recognition sequence. So contains monomer and heptamer separated by 12 base pair and 23 base pair. So it's so-called the 1223 DNA. And the very top is the coding DNA, which is coding B or D or J, having no sequence specificity. And active center is cleaving it here. It's ready to make a hairpin on the top, blunt end at the bottom. So what you notice is the 9 base pair conserved sequence in the two different DNA, they're almost anti-parallel. Monomer yellow here, the monomer on the 23 RSS is pointing into the board. But the coding flank and the heptamer 7 base conserved sequence is almost parallel. So from anti-parallel to parallel, and it requires a lot of bending here, make it very asymmetric. And the protein is pretty smart, many domains, interdigital crisscrossing binding the two different DNA. And they cross over 3 or 4 times along the way. So to tie the DNA together, so only 12 and 23 will be paired. And it kind of reminds me of the Asian tradition at wedding, groom and the bride have to crisscross their arms to make sure that in lifetime they will be bound together. So that adds the active side domain and the RAC2, so you can see protein mostly running in between two DNAs to tie the two together. So now, I told you we started from crystal structure selenomat from the beginning. Then we get the protein DNA complex. This is the really fine-tune the DNA sequence and make the specific DNA substrate so it's ready to make hairpin. And we get to this 2.75 Armstrong. But when we provide the DNA to the protein, the DNA is uncut. We ask the protein to make a complex to make the first nick. Somehow the DNA was just not positioned right for nicking. I'll show you more detail. And all those were crystal structures before 2017. And then we have to use cryo-EM to get the step that's DNA is being nicked by RAC2. And that required cryo-EM. I will tell you why and how we got it. So basically, when we provide the DNA, it's fully intact, and this is the cysyl phosphate, red, supposedly should be put in the active center defined by those carboxylate, but it's 180 degrees away, and the active site without cysyl phosphate in there the third phosphate, E962, is far away from the other carboxylate. So not only it needs to be a 180-degree rotation, this is also a little bit short, right? Vertical distance along the y-axis is short. We know it has to happen because it does nick. But in the structure, even in crown EM structure, initially we cannot get it. And in order to do it, we would have to go through mutation in the DNA. We introduce a basic site, removing a base here, you can see there's no base. And then more mutations in the lower portion showing magenta to really force it from beginning we have 0% rotated the molecule. And then this is 0%, doesn't matter, thermodynamic energy to force DNA to unwind by 180 degrees, or with one mutation, we can see we have increasing amount of 180 degree rotated one. But when we make 3 mutations, even at room temperature, we can see 100% unwound. And I'll show you how the DNA is unwound because it forms a new conformation never really seen before. So the DNA unwound by 180 degrees. And this happened naturally when it's bound to the protein and form this we call a zipper. So it's unwound 180 degree, 2 DNA strands parallel rather than coil. And then the base stuck in the middle form this driven by the base-base interaction stacking. And then we have this conformation that we also collaborate with Robert Best, who is in MR building 1 floor above us, and he proved that by energy-wise is the most stable conformation. And not only it's unwound 180 degree, it pushed the cis-phosphate right into the active site. And the carboxylates are now all aligned with 2 diagonal cations ready for cleavage. And I have to say, when I was a graduate student, my advisor said, when you first observe something, you think you discover something, be careful, you may be wrong, because it's never been observed before. Who knows it's right or wrong, right? So we were nervous. Besides, we asked Robert to do the simulation. We looked into it, just happened to be just the same year when we observed the structure. Osha Barbas in Anbold and Heidelberg reported a structure of another protein bound to DNA. In her structure, the DNA is also unwound. It's a crystal structure, even though it's unwound by only 60 degrees. Nevertheless, it formed a similar zipper structure. And the zipper requires purine pyrimidine alternate, TA in her case and the CA in our case. So the purines in the 2 base stack gets stacked together. So now I'm just going to put together, we have crown structure combined with protein mutations, we get this zipper structure for DNA nicking. And then we also have crystal structure for hairpin formation. So we put together a little movie, how it works. So this is initially how protein binds with DNA look like. An active site is down here. The first bound to be nicked is up there, cis-phosphate. The second one to be cleaved is here. Even though it's initially close to the active center, no matter what we do, it can never be cleaved because the active site is not fully formed. And so undergo this zipper formation, 180 degree unwound, so it can nick the DNA. After nicking, there's a large conformational change. I'm only showing you around the active center for the hairpin formation, but it's globally there's a structural change in the ribonucleic DNA complex. So this is how dynamic DNA is, and I say DNA is not so boring. It's not always double helix. It could be different. And given the special sequence in this case is CACATGT. So basically, whenever you see pyrimidine purine CATG, it's prone for deformation, unwinding, tilting, bending, etc. And when we superimpose the active center, now we have both reaction steps. The carboxylates and the protein can be superimposed really well. But the third carboxylate shown here actually have 2 conformations depending on the reaction and need to capitalize. And the cis-phosphate is really well superlaid. The yellow strand is first strand to be nicked, and you can see it's one directional of the DNA going. And the second strand needs to be cleaved, going from opposite direction. But then it bends 90 degrees, so the cis-phosphate is perfectly superimposed. And the nucleophile, one reaction going from water, going from right to left, and for hairpin formation, the nucleophile 3' OH is going left to right. And so the same active center can catalyze two types of reaction, and the reaction is pretty much conserved. And except which nucleophile, it's like a ping pong reaction. So with the remaining few minutes, I'll switch here, moving from making the hairpin to make an antibody-antigen receptor to how our cell able to open the hairpin before it's ligating to make final product. And for open the hairpin, there's a specific enzyme in our body called Artemis, which is a hunter. And Artemis is a finicky nuclease like this one. And it needs a lot of things to coax it and help it. It needs something called the DNA PK, DNA-dependent protein kinase. And Q70Q80 are the ring-like, topologically close to ring-like protein needed to form the DNA PK. So catalytic subunit plus Q70Q80 have to find the DNA end, then recruit the Artemis to open it. So my lab started to work on DNA PK in 2008, and at the time the year was still crystallography. And DNA PKCS, the catalytic subunit, is 4,128 residue in human, and it's huge. And the only way we can purify this protein is from helical nuclear extract, say native source. And Joyce was a postdoc then, and she was really capable. She was able to make Q70Q80 to heterodimer plus DNA PKCS, and assemble with DNA for crystal formation. And you can see this is the structure eventually solved using cryo-EM. Q70Q80 is pinkish. The DNA PKCS have 4 domains, we say blue, N-terminal heat, N-heat, green, middle heat, a lot of heat repeats. And so the green and blue form this sort of pincer to grip the DNA. And the top is the fat king kinase, so the yellow and orange. And so the crystallography, Joyce grew the crystal, but never diffract so well, and she dissolved the crystal, regrew the crystal using crystallization and purification. She got to nearly 5 angstrom, but it's just nowhere enough. And the Tom Brundel's group was super. They were able to solve the structure, the best 4.85 angstrom published in 2017. So I told you we made the holo enzyme on DNA, but it was waiting. When the cryo-EM revolution happened, we basically hopped onto the train and moved. Structure was solved. The initial structure was solved at it's a 3.7, the best portion 3.2, worst portion is 4 and 5 angstrom. So we cannot be sure the structure is absolutely right. So what happened is we used the selenium mat reported by Tom Brundle to solve the crystal structure. And we basically use a selenium mat position to help us to build the model. And I have to say 4.85 angstrom crystal structure have errors. Overall structure is right, but misalignment, etc. was severe. So basically, Xuanming group was able to use their structure selenium mat position to build our model. And then he went back to use our model to re-refine the crystal structure. After he finished, done. So the crystal structure R value, both R and R3, was improved more than 10%, both 10 or 12%. And the geometry was also significantly improved. So basically, cryo-EM structure can improve the low resolution crystal structure and vice versa. So I think it's a lesson of what we can do in addition to AlphaFold, because after all, selenium mat is real experimental data. So the protein is very, very mobile, from able to DNA bond to Q70Q80. It's enormous. So I'll just focus on one point. It's a lot of things to learn from thousands of amino acids. What's really interesting of DNA PKCS, it's a kinase, protein kinase, phosphorylating threonine or serine. And the most important thing is that it phosphorylates itself. The self-phosphorylation site, one site is called ABCDE, cluster phosphorylation site. And if the mouse doesn't have DNA PKCS knockout completely, mice were born, and it's okay. And they develop okay, only they're a little bit sensitive to radiation, gamma radiation. But if you knock out either the phosphorylation site or make the kinase dead, then the mouse is embryologically, so couldn't survive. So it just shows how important phosphorylation is. And it's called a DNA-dependent protein kinase because only in the presence of DNA it has kinase activity. So with the structure, we can explain how does that happen. First of all, without DNA, that's Tampere structure, the portion that needs to be phosphorylated is shown in red. It's inside the green ring. And the phosphorylation site is the kinase on top, like at the head. So they are far away and its trapped, cannot be phosphorylated. Once we added DNA, what happened is that we found this particular patch become external of the ring and it put next to the kinase. But we cannot put ATP or ATP analog in it because once we put in that, the structure is very heterogeneous. And again, that's like a sort of technical tool. What we do is that then we know Artemis depend on this DNA PK to open the DNA hairpin. And in cryo-EM is the more the better. Not only the more the better, you learn biology better. A lot of time we have preferred orientation issue, but if you enlarge the protein, adding more component, adding the disordered region, somehow the preferred orientation issue can resolve itself. So in this case, when we don't have the Artemis, the compost is very had preferred orientation issue. Once the Artemis added, it was very good. But more than that, there's the key. I told you if it's phosphorylate itself, it becomes released DNA, and it's become very heterogeneous. So what we solved the problem is by following the kinetics. The red line is kinase self-autophosphorylation rate. You can see it gets fully autophosphorylated by 10-12 minutes. But by the Artemis cleavage, opening the hairpin showing the blue line, it's very slow. Even an hour later, it's only 40% or less DNA is open. So it gives us a window, microgroup like to do time course in crystal or in cryo-EM. So because the big time difference, so we did the ATP incubation. Without Artemis, we do one minute at most manually. We can freeze the grids, and then we can trap the phosphorylation state. With Artemis, the life is much longer. 4 minutes, 8 minutes, 10 minutes, we all can get autophosphorylated form, yet Artemis hasn't cut the DNA. So I will just show you that we had a structure of the Artemis, not Artemis, basically DNA PK autophosphorylate itself. It's kind of interesting. People talk about post-translational modification. A lot of times we hear phosphorylation is utilized as like a fishing hook, so basically picking up other DNA or other protein. In this case, the phosphorylation site is auto, and the binding site is within itself. So basically, there's a multiple of 4 phosphorylation sites identified, shown here, 1 and 2, 3, and 4. And what they do is that the top is the kinase, the phosphorylation peptide wrap around this green middle heat. 1 is on top of the middle heat, 3 on the bottom. What it does, those binding sites has never been known before, but structurally we find the site and it basically lift the middle ring. And basically, we find all those binding sites, and biologically we can test the function in collaboration testing MIC. That I told you, if we mutate all the phosphorylation sites to the playation 3 with alanine, protein lost the function. Basically, wild type is this much function, and the mutant is that function. Then we mutating the surrounding hydrophobic residue, which is important for this binding to occur, it's also lost the function. And then further, if we mutate the lysine, which is interacting with phosphate, same deal. So basically, phosphorylation is essentially to form a self-interaction and remodel the protein. So here's the movie, how the structure changed. So the front view, you can see this gigantic amount of change. Okay, it's the first time I saw phosphorylation site can remodel itself, the protein itself. And this the front view, and then the side view is the other one. And you can see when I say about lifting the green NP green, the binding phosphorylation binding site is on top of here, it's shown here. And then DNA binding occurs at the bottom, so basically opens DNA binding. And this heat repeat occurs in many, many proteins. People always say, why are there so many heat repeats, helical repeats? It reminds me of the slinky motion. It's not simply domain movement or helix unwinding. Helix can twist, bend, expand in different ways. So basically, it's underlying many structural conformational change. I don't think AlphaFold can predict that yet. Anyway, so the phosphorylation lead to the DNA binding groove open up, allow DNA to be released. But the problem is that if the DNA is released, it's not good news for processing. And the Artemis actually gets inserted in between the open DNA binding grooves, and that get a hold to open the hairpin. So it's choreography of DNA end, moving in, out, protein opens, and so the nucleus can be inserted. So the Artemis has the nucleus domain, which is inserted in between DNA and this blue, the green MP, and then it has a long neck hanging up onto the near the kinase domain. So I have this phosphorylation dependent conformational change allow the nucleus to access DNA end. So I'll end my talk here because I think the interesting part is how to get all those different states. And the work is supported by NIH and the long-term collaboration with Marty Gellert, who is still working. And the benefit of the NIH is you can work to any age to your heart's delight. Marty's way over 80. And it's still very active. And the people involved in this work, I mentioned them along the way. So thank you for your attention.